Good morning, everybody, and welcome to Osher Lifelong Learning at UCSD. I'm Mavis Porter. I'm a member of the Curriculum Committee. I feel like Lindsay gave such an impressive introduction that I don't need to say a word. However, I'll say a few. Today, I'd like to welcome Professor Carl Marchetto, which I hope I pronounced properly. Carol is Assistant Professor of Anthropology at UCSD. Prior to her appointment, she was a staff scientist in the Laboratory of Genetics at the Salk Institute, where she led projects that studied neurons, neuronal development using human and non-human primate stem cells. She earned her BS in biology, biological science, and a PhD in microbiology from the University of São Paulo, Brazil. I'm sorry, there's a light in my eyes and it's really not easy to see sometimes. So welcome to Carol Marchetto. Thank you.
Is it better now?
Yeah.
Okay. Also, I think if I speak towards the microphone, it will be better. Okay, great. We will work with that. So today I'm going to talk about the use of stem cells, which is a technology. It's a tool to study human evolution, human origins. So we're starting there with a comparison of a human, about two humans and two chimps. Chimpanzees and bonobos are the closest living, our closest living relatives. And the idea of this image was to tell you that we actually have much more in common than we have differences, even though we feel that we are so different from chimpanzees and bonobos. But when you look at, for instance, DNA evidence, we are, depending on the analysis that you look, about over 98% similar to chimpanzee and bonobos. So we're very similar. So what are the differences and how can we leverage some of the new technologies that we have, such as stem cells, to study some of the differences? But first, I wanted to tell you what are stem cells. Stem cells are cells that are very special. They have two main characteristics. They can self-renewal, which means they can make more of themselves, technically forever, in culture. Also, stem cells have this property, very important property, called pluripotency, which means that they have many potencies. They can make many other types of cells other than themselves. So you have these very special cells that can make more of itself forever, and it can also make, in theory too, all the cells in the body. So those are quite cool cells. And up until not a long time ago, let's say 98, the technology, 1998, the technology of culturing, having those cells in the dish so we can work with them and understand some properties of stem cell, as well as generating the other cell lines that the stem cell can generate, that was not possible. And just recently, we started working with those stem cells in laboratory setting. So how do we find them? Where they are originally in our body? So the stem cells in our body are very, especially the embryonic stem cells, which are the ones that have a lot of potencies. They can pretty much become anything in our body, other cell line in our body. They are found very briefly in the blastocyst of embryo. So this later, so after egg is fertilized by sperm cell, there are some divisions that happen, and not to get too much into embryology, one of the stages of the development of the embryo generates this structure called the blastocyst, which is just a bunch of cells organized in some specific way. And at that stage, that's when in clinics of in vitro fertilization. That's when the embryo is implanted in woman for pregnancy. And when those cells are not looking good, the clinic will discard the lines. And if you look at this area called the inner cell mass of the blastocyst, those cells have this pluripotency. So they are the ones that will become your whole body. So they're quite special, these cells. And during mammal development, these cells just exist for a little bit, and then they will, of course, specialize and differentiate into different cell lines. So what can be done is, at the time where these cells are present, those cells can be cultured in the dish, and you can study the properties of self-renewal and pluripotency in the dish. So those are the stem cells. And why what are some considerations about that? So, of course, to use these cells, especially for humans, you need to obtain cells at the blastula stage, which is the pre-implantation stage. And at that stage, there are some ethical considerations about that. And it's also challenging to obtain embryos from humans, and virtually impossible to obtain embryos from apes. And we would still like to understand how, in terms of looking for differences between humans and chimpanzees, for instance, and we're going to be referring to them as non-human primates. We want to know how a stem cell become a neuron, how a stem cell become a brain cell. So that window in neuronal and brain development can be really important to understand some aspects of how we can do such complex brain processes the way we do, and comparably, chimpanzees, bonobos, and other non-human primates, they can't, even though we still do a lot of things similar. But there are some things that our brain does, that the other brains from non-human primates don't. So understanding brain development using those cells can be a great important tool. So what is an alternative of taking the cells from the inner cell mass of the blastocysts without touching the embryo? So about some years ago now, around in 2006 and 2007, a researcher called Shinya Yamanaka from Japan. And later on, this discovery was so important that he got a Nobel Prize for that in 2012. He discovered that if you take skin cells, for instance, and now we also know that we can do that with blood cells and other types of cells that are more readily accessible, you can change the program of these cells and trick them into becoming a stem cell. So the thing was so simple that, and he added a few factors to, I'm showing here skin cells, so those are the skin cells here. Do you see my cursor? So this researcher very simply cultured with his postdoc at the time, cultured fibroblasts, which are one type of skin cell, and used a cocktail of literally 4 reprogramming factors that he called reprogramming factors, because those factors were able to change the fate of this fibroblast cell that thinks is going to be a skin into a stem cell. So it's kind of like turning the clock of development back in time. So now, these fibroblasts, after being exposed to these reprogramming factors, now behaves just like a stem cell. Of course, it doesn't take 2 days. It takes about 6 months to a year to do that. Now we're getting a little bit faster, so about 4 to 6 months. But at the end of the day, you can choose an individual or a non-human primate. If you have access to a skin or blood from that individual, being that individual human or non-human primate, you can have stem cells from that individual. So that allowed us for the first time to study neuronal development from living individuals without, of course, having to break their heads and look at what the brain was doing, which is something obviously we don't want to do. So because it's so hard to assess brain function because it's so well protected, it's a very important organ, we use this type of technology to now look at brain cells from living individuals in the dish. And he called that, Shinya Yamanaka, induced pluripotent stem cells, because they were not stem cells to begin with, but they were induced to become pluripotent. So the way for short, you see a lot of IPS, which is induced pluripotent stem cells. So what are the implications and advantages? So there is no need to assess embryos to obtain stem cells anymore. Sometimes we still use them as benchmarks to make sure the cells that we made are doing what they're supposed to do. But pretty much a lot of the work on stem cells, especially this type of pluripotent stem cells, is done with only this technology of inducing directly from the skin. So in terms of evolution, this allows us now to study live cells from primates that are not easier or ethically accessible otherwise. So there is no way you can look at live neurons from a person, because you would have to go through the skull and look at that brain and find the neuron and image that, which is not possible. And the same for non-human primates, especially apes. So now we can do that. For mental health, that has obviously big implications too, because now you have this platform to test hypotheses. You can study cells, for instance, from living individuals or patients that have either a developmental, a psychiatric condition, or even a neurodegenerative condition. And you can genetically modify these cells to study and ask questions. For instance, you can have siblings that one has a condition, the other one doesn't. And you can compare cells, brain cells from those two individuals, and you can see what the neurons from the individual that has a certain neurological condition is doing that the other one is not, or vice versa. So you can get, you can have this platform to ask some of those questions related to mechanistic changes that we would not otherwise, because we can access the brain. So how can we use this STEM technology to ask questions about evolution? So more specifically, before this technology was accessible, the way in which there were still models to study human evolution, and the way in which we used to do that is we would look at DNA evidence, so we do genetically comparisons. Remember I said humans and chimpanzees are very similar. What are the differences? So you can look at the DNA and see what are the different genes. Genes are pieces of DNA that codes for RNA and proteins that makes the cell function, so you can compare those. And we used to do that before, so you can get some information. You would look at postmortem tissue, so you wait until the person or the individual, either human or non-human primate, dies, and then you look at their brains and you see a picture of that moment in time, right? So that's good, but it doesn't really give you live information of what the neuron is doing. It just gives you a picture of what the neuron was doing at that time, at that point in time. You can also look at the fossil records, and that can give you an idea of how this organism interacted with other organisms and the environment. That can give you some information, of course, but it would not tell what the neuron is doing still, right? And you can also look at archaeological evidence. So that were the models that were present before this technology. Now, we feel, pun intended, that there was a missing link, and that missing link is the possibility of study live cells. So now with IPS technology, you can find ways to integrate all the other tools that were already available to study human evolution, and now we also have information about living neurons. What are they doing? Or the neuroprogenitors, which are the baby neurons that are going to become neurons. So how this development happens. And that's what we did. We know that there are differences. Again, we're so similar, but we also are different. So chimpanzees and bonobos, what are the differences? They share about 97 and 98% of our genome, chimpanzees and bonobos. We know that there are differences in brain size. There are differences in culture, differences in language. We know that there are also differences in susceptibility to infections, susceptibility to neurological diseases. We wanted to see if it was possible to use that platform to identify cellular and molecular differences between human and non-human primates that would potentially improve our understanding of the evolution and diversity of our own species. So how did we go about that? We took skin samples from human, bonobos, chimpanzees, gorillas, and rhesus macaque. And some of those samples came from our very neighbor, the San Diego Zoo, which has a beautiful colony of bonobos. So then what we did with those cells, and we call them somatic cells, which are the cells from the body that are not pluripotent. Those are fibroblasts in that case. And we use the reprogramming factors that Yamanaka already had published before. And we made induced pluripotent stem cells from them. And the idea is we wanted to compare the differences between neurons from humans and neurons from non-human primates. So first question was, are there differences that are detectable at the cellular level that will be relevant to understanding human brain size and function? We didn't even know that for sure, because maybe what if when you take the cells away from the body, the cells don't show differences anymore? So we call that, when there are differences even outside the body, we call that cell autonomous. So that means you can identify a chimpanzee cell no matter where you put it. So that was our hypothesis going into this work, thinking that, okay, we will be able to detect differences at the cellular level, even outside of the body. So as I said before, we first made iPS cells, and that's a lot of images here, but just so you can see how this is how a fibroblast look like under the microscope on the bright field, and this is how induced pluripotent stem cell looks like under the microscope. So that's you can hopefully maybe be able to see isolated cells here. So the fibroblasts are spindly and grow very flat. So those that will be, let me see if again. This will be one cell, for instance. And you can't quite see the nucleus, but you see the whole cell. And when you look at induced pluripotent stem cells, they grow very differently. They grow in colonies. So when you look at this guy here, this is almost a whole colony. You can see the edges, the borders. Here is a part of a colony too. This is another part of the colony. And here you can, I'm going to put my cursor on one cell. See this little round thing here? Oh, it's hard to see. But it will be easier to see here. See the green dots? The green dots are showing us stem cells. So that's a colony of many green dots, and those are all stem cells. So how crazy is that we can make these guys look like these guys with only 4 factor and a lot of time. But it can be done. So that's an important technology. And we did all the controls to make sure that they were stem cells, they were doing what they were supposed to do, and their karyotypes were stable. Because sometimes when you mess up with cells too much, you worry that they become, they get a little bit messed up and they don't have the karyotypes stable anymore. But in that case, they were. And you can see humans have 2n equals 46, as we all know, and chimpanzees and bonobos have 2n equals 48. So we looked at that. Then the next question is, we don't want to spend too much time working on the induced pluripotent stem cells. We want to look at what's happening at the neuronal progenitors, which are the young neurons, and we want to know what's happening at the neuronal level. So first thing we did is we subjected this, and that's a protocol that we've been doing forever. Not important to know the details, but with an addition of different growth factors, we can guide the cells that are pluripotent into start differentiating into neuroprogenitors, for instance, which are the baby neurons. And here are how they look like. They go through that phase where they become, we call neural rosettes, which resembles a neural tube, the developing mammal neural tube. And then we dissociate the cells and plate them into specific conditions, and they grow like so. And we call that population, we can work with that population, pass, expand, and bank. That's our neuroprogenitor cell population, or NPC, as it's shown there. So once we had that population, because we didn't really know where to look for differences, we took first an unbiased approach. So we took populations from humans, of human neuroprogenitors, chimpanzees, and bonobos, and we wanted to, our strategy was to use high-throughput RNA sequencing. So that's when we don't really know what to look for, so we just asked, what is it that this cell is making? And in terms of, and cell makes RNA, and then protein. So the DNA is transcribed into RNAs, and RNA is made into protein. And the way in which we, one of the ways to compare is to look at all the RNA that the cell is making. So that will tell you what the cell is going to do, or what the cell is currently doing. And there are many things that the cell are doing at the same time. But because those are neuroprogenitor cells, a lot of what they're doing is the same, but we wanted to see if there were any differences. So to give you an idea, and we call it gene expression, because RNA is an expression of what the genome is doing at that given time. So, and then we thought, okay, if we see differences, we will look at assays that can determine whether the expression differences are reflected in a function or the behavior of the cells that we are looking. So we looked at that neuroprogenitor population that I showed you before. So how does that look like? So when we looked at everything that the cell was doing in terms of RNA, so that's RNA expression, the expression of the genes into RNA, and we saw that over 35,700 genes were the same. And that makes sense, right? Because human and chimpanzees and bonobos are very similar in terms of the DNA composition. And a neuroprogenitor cell is a neuroprogenitor cell. So they're all doing very similar things. So it was not unexpected that there will be a lot of similarities. And in fact, there are. However, when you compare Homo, which is for Homo sapiens for us, and Pan, which is for Pan troglodytes, which is chimpanzees, and Pan penices, which are bonobos. So when you compare them, you can see that chimpanzees and bonobos expressed some proteins and some RNA that were specifically for them, and same for Homo. So when we sum the amount of differentially expressed genes, we noticed that there was 1,196 genes that were differentially regulated between human and non-human primates. So and then we started doing analysis to see which were the categories in which those genes will fall into. And what we noticed is that 52 genes were involved in cell migration. So we thought, okay, now we have a lead that maybe to look for some differences in cell migration. How do we do that? So we focused on experiments that would help us to maybe see cell migration. So what did we do? We did our first experiment was a quite simple one. One way to see migrating cells is to make a scratch in the dish and look at the cells migrating towards closing the scratch. It's a quite common phenomenon that when you have a scratch in the dish, the cells will migrate towards the scratch and will close the gap given some time. So we did some, so that's, oopsie. Wait, let me try here. Oh, yeah. So that's the phenomena. The phenomena is given a scratch in the dish, the cells migrate towards the scratch and close the gap given time. So that's a time lapse, what we call, which is, it looks like a video, but are many, many images taken over time. And when you play all those images together in a microscope, in a computer, after you're taking the images in the microscope, you can see the video of the cells closing. And then what we did is, because we wanted to assess speeds, we wanted to assess the speed of the individual cells, because we thought migration was important too. We also added a fluorescent dye to the cells so we could track individual cells. And let's play. Okay. So here for instance, purple and blue and yellow, you can see us tracking the cells. So we tracked individual cells for the time of the closing of the gap closure. So now we could get speed and direction as different parameters for the migration. So a very simple analysis over time showed us that human cells so given a same scratch in a human dish or a dish filled with chimpanzee NPCs, in 24 hours later, chimpanzee lines had already closed the gap, and human lines have not closed the gap yet. So we did that many, many times to get numbers and to make sure that our observation was real. And then we indeed observed that in this experimental parameter, chimpanzees and bonobo cells close the gap faster than humans. So chimpanzees and bonobos are faster. So of course, we wanted to look at other experiments to see if we would see the same thing, right? Just as we do in science sometimes, we make an observation and then we want to see, okay, if that's really the case, if we raise the cells, if we put a human cell and a chimpanzee cell together, who would win? You would expect that chimpanzees would win, right? If I could raise the cells. In fact, there is a way in which we can add the cells in a plate. And this is a special type of cell culture plate where you have, so those are the cells here, and we color-coded them to make it easier to know who is who. We plated them on one side of the dish, and in between there is a 5 micrometer, so a long stretch, where the cells are constrained to those little grooves. Those are the grooves, and only one cell can go through at a time. And the way in which we added the media, there is the cells will migrate to the other side. There is a gradient push for the cells to migrate to the other side. And we wanted to see which cells will end up on the other side of the microgrooves. So here on the left is how the microgroove cells, the microgroove plate looks. So those are the little grooves. And here is an example of a cell squishing through a microgroove. So only one cell can go through. And here below is where the cells are going to migrate. Cells migrating towards the top. So the migration goes that way. So let me play a video just so you can appreciate how the migration happens. So cells are migrating towards the top. And because there's a lot of grooves and it's kind of hard to see who is migrating where in terms of reds or green, I will play only the fluorescence. As soon as it stops, okay. And then you will tell me which species is green and which species is red. I know if it wasn't, so doesn't happen. So who do you guys think is green and who do you guys think is red? I know, I mean, we've done that a lot of times. So that's an example of, a representative example of what we have. So the red cells are faster than the green cells. And we see that in general, even with the other lines. But point here being is that the red cells are the chimp cells. So over time, we observe that when we raise the cells, chimpanzees and bonobos are still faster than humans. So now we are looking at another paradigm, experimental paradigm, that is showing the same result. That's always good in science, when we look at different paradigms that show same result. So in fact, the chimpanzees, and it's also true for bonobos, cells migrate faster than human cells. And here we show a quantification of the different speeds. And you can, I think I covered the right side, the axis here. It's micrometers per minute. So that's the different speeds. And you can see, amazingly, that chimpanzees and bonobo lines are quite comparable. So that's telling us that that's something intrinsic to the cell, that even if the cell is outside the body, you still see those changes. So that's quite, that was quite impressive for us. Let me quickly look at the time. Oh, I have it here. Okay. So what about neuronal maturation? We looked at the neuroprogenitors, which are the baby neurons, the young neurons that are going to become more mature neurons. But we know that as the brain develops, there is less and less progenitors and more mature neurons. So we wanted to see if we could see some differences at neuronal neurons that were more mature. So we wanted to look at this population here, the neurons. So we use the same paradigm that we use to make the neuroprogenitor cells, and then we push them a little bit further so then they will become more differentiated neurons. So every neuroprogenitor cell now can have processes, extend processes, communicate to each other, and something that the neurons do that it's quite typical from neurons is they fire action potential. So one of the ways in which they communicate through each other is through neurotransmitter and electrical activity. So first, we just wanted to make sure that we were making neurons from all the species, and in terms of general markers for neurons, so the neurons were looking the same. So which is good because humans and non-human primates have neurons. So in the dish, it's the same thing. We can make neurons, and the proportions of the different types of neurons that we can generate in culture are the same in humans and non-human primates. So that's a good control for us to know that we can make neurons the same way for humans and non-human primates. But then are there still differences? And to look at differences in neuronal activity, we used another technique called a platform called the multi-well electrode array, or MEA for short. So what that is, we have those plates. There are special plates, and inside those plates, every well had a set of electrodes, and those are here the ends of the electrodes embedded in the plate. And we will plate the cells on top, the neurons on top of those electrodes. So those electrodes, and here is, if you look at a cross-section of the plate, a scheme. So here will be an electrode, and we'll plate the neurons on top. And the idea is that the cells that are cultured directly on the top of the electrodes, they will be able to, if they are forming a network, they will fire action potentials, and the electrodes will detect extracellular changes in voltage. We also call that spikes. And here you can see how in live imaging that looks. So that what we see here is this will be the equivalent of one well. And here you can see one neuron is firing, and then another neuron is firing right after. After this one firing, another one will fire here. And so we can quantify these events and have an idea of how active the cells are, because they might be firing more or less action potentials, or spiking more or less. And we can also learn how connected they are, right? Because if one neuron is firing and the next one close by fires too, that proximity in time of firing can indicate good or poor connectivity within the plate. So what we do is we plate human cells in many wells, and we plate non-human primate cells in many wells, and we look at the output. So the way in which this analysis come out of the machine, so the output allows us, so there will be per channel, you can see those traces, and every trace will be correlated with a spike. So every time there is a trace, it means that the neuron fire. And what we can do is, over time, we can count the number of spikes, and we can look at the time where the spikes fire all together, so that will be a neuronal burst. But we can also look with the other channels how many times the spikes happened synchrony, in synchrony, which means there is connectivity within the plate. So how does it look like? So early on during differentiation, and here we're looking, so human neurons and same for chimpanzee neurons from neuroprogenitor cells take up to 8 weeks to get more mature. So right now in culture, so we add all these maturation factors for the neuron to become active, and we look over time, because we can take the same plate and measure in different time points during development, during neuronal development, so you can follow the same the same cells and see how they progress over time, right? So in 2 weeks, and that's important because changes in maturation sometimes are related to changes in brain development. What I'm showing here is an example of a human well, and an example of a chimpanzee and bonobo. And in 2 weeks, there is not that much activity. We're looking at all the channels, and which are equivalent to all the electrodes that are embedded in the plate. So there are 8 to 9. We're looking at 9 here. And we can see that there is some activity on channel 2, some activity on channel 6, and very little activity on channel 4, right? When you look at chimpanzee and bonobo, clearly we start already seeing more activity than we see in humans. And that's a significantly difference between, and here I'm graphing those firing rates. It's called mean firing rate. We're looking at hertz. And when you look at human activity at those early time points between 1 and 2 weeks, humans are less active than chimpanzees and bonobos. What happens at 6 weeks post-differentiation is that now human cells have many more channels active, but not only that, they have more synchronicity and more bursts. So both synchronicity, which are the pink, we call the network bursts, which means that all the lines are firing, all the channels are firing at the same time. And we also have more bursts. So bursts are the neurons are firing more spikes at a given time. So those are hallmarks of increased maturity. So 6 weeks later, human cells have increased maturity, going up here, compared to chimpanzees and bonobos. So what we conclude here, is that we see accelerated differentiation of chimpanzee neurons, and we see protracted development of human neurons. So again, chimpanzees and bonobos seem to be accelerating compared to humans. So there are some theories that talk about that. In human evolution, protracted development, or this delay of more mature features, also called neoteny, it has been proposed as a possible mechanism that contributed to the rise of the human-specific features, including an increase in brain size and the emergence of human-specific cognitive traits. That's the idea that if you rush through development, you could compromise proper maturation. And proper maturation in human neurons can mean many things, but one of the things that it can mean is proper pruning. So just like a plant, when you're when you have a plant growing in your garden that you don't prune properly, the way in which it grows might not be beneficial to its own health, and the way in which neurons grow in the brain goes through a process that is like that too. Non-human primates, all the neurons go through pruning, but pruning takes a lot of time. So that delayed maturation makes us very useless when we are babies, but can allow potentially for us to have a more stable and carefully crafted architecture in terms of how our neurons integrate in the system. So that's this idea of neoteny. So really quickly, and I'm going to get a little bit more specific now, a remaining question. So that's giving you a little bit of food for thought of what were the remaining questions that I will address at the CARTA meeting.
November 19th.
So one remaining question is how neoteny is regulated, because we see that in our cells, but what is happening inside the cell that could be contributing for this process? Because now we're just describing a phenomena, and we want now to try to explain why that happens. So to do that, we took, again, this unbiased approach, and here we looked at how the RNA, again, the high-throughput sequencing RNA, we looked at all the RNAs from all the human and non-human primates, and here we added chimpanzees, bonobos, gorillas, and rhesus as an outer group. So rhesus are not apes, they're monkeys, so we want to compare them with the other non-human primates. And here we call principal component analysis, and without getting too many details, because I'm getting to the end of my talk, it's a way in which you can see, you reduce all the RNA sequencing genes that are different in one little dot. So it's a way in which you can visualize all your cell lines, and you can capture the variability, but you only see one dot per sample. So what this is showing you is, when I look at the expression profiles from neuroprogenitors and neurons, there is a difference. So that's known, right? So neuroprogenitors are baby neurons, and the neurons are more differentiated and have this differentiated feature. So that's when we call the principal component 1. It does explain 28% of the variance. Now, when we look at the principal component 2, we can see that the cells separate in phylogeny, in terms of phylogeny, which is really interesting to see too. That's also telling us that if I have an isolated cell from rhesus, macaque, it still clusters separately, both neuroprogenitors and neurons, and more distant. So remember, it's an outer group, so it's clustering more distant than the apes. And gorilla now is, the cells are clustering closer to human and chimpanzees and bonobos. But now chimpanzees and bonobos here in pink are very close to humans. So we are reproducing what we have in phylogeny in this graph, which is important. But most importantly, we started finding specific genes that were clearly differentially regulated in human compared to non-human primates. And an example here is transcription factor. Transcription factors are genes that, especially the pioneer transcription factors, they're very important because they find places in the DNA, they bind to many areas in the DNA, and they have very important, strong downstream effects. So that means, for instance, transcription factors are what Yamanaka, back in the beginning of my talk, used to do the reprogramming. Because so transcription factors are so important and can make so much change in the cell that the factors used by Yamanaka were transcription factors. So that's how important and strong effect they can have. They can change a fate from a fibroblast into induced pluripotent stem cells. So GATA3 is one of those examples. He's not one of the ones that Yamanaka used for reprogramming, but GATA3 is a factor important in cellular differentiation. And it's lower expressed in over the course of differentiation. So 0 here, it's neuroprogenitor cells. So it's lower expressed here. And it stays low for all the other non-human primates. Bonobos, chimpanzees, gorillas, and rhesus. And in humans, it goes up right at the time when you start the differentiation. So that told us, and it's so significantly changed that it's at the top of the list of the differentially expressed genes. So we thought, okay, let's look into that. And in fact, when you look at the protein levels, it's present in humans, and it's absent in chimp, bonobo, gorilla, and rhesus. And those are the control lines. There are human lines that will show expression. So, and we also know from areas in the brain, when you look at the postmortem atlas made by the Allen group, we look at the areas in the brain that would have GATA3 expression. We don't have chimpanzees there yet, but it's a bank that has postmortem brain tissue from developing brains. And when we look at rhesus macaque, we don't see expression of GATA3. So low expression of GATA3 in rhesus, high expression in human, which is correlating to what we see in vitro. Okay, so real quickly what we did a quick experiment where we decreased, if GATA3 was important for neuronal maturation in humans, what happens if we remove GATA3 from human cells? So that's what we did. We used a trick called shRNA, so that's a short hairpin RNA. It eliminates or decreases the expression of a certain gene. So now we were able to have human cells that don't have that gene. So what happens, so that's just to remind us, right? Human was slow differentiation compared to maturation compared to chimpanzees and bonobos, right? So what happens now is with this mean firing rate is if we compare humans, low here, with that's just an empty vector, but the important point here to show is that top one. So when we remove GATA3, now we have a cell that is more similar to chimpanzee or bonobo, and it has more firing. So by removing GATA3, we managed to push human neurons to fire faster. So we think that we're starting to find some of the important mechanisms by which neoteny happens in humans. So decreasing expression of GATA3 gene in human neurons increases neuronal activity to similar level as chimpanzees. So that gene is definitely one of the genes that is important in this neoteny phenomena that we see in humans. And there is more to come, hopefully, in the future. So that's what we notice here. So conclusions for my work, our work here is human neuroprogenitor cells migrate slower than chimpanzee and bonobos, both in vitro and in vivo. I didn't show the in vivo data. Human neurons show slower maturation, both in morphology and function in early development. That's what we've seen, that humans are slow in terms of firing in the beginning. Human neurons show evidence for neoteny, and the gene GATA3 is involved in regulating neoteny in human cells. So we think that this work together show that there is evidence for cellular features that are totally independent from the body that will distinguish human from non-human primates, and they are preserved even outside the organism. And that's important because it means we can use this model, right? So we propose that the primate-induced pluripotent stem cells can be used as a model to study dynamic developmental comparative differences between human and non-human primate. And those differences could have an evolutionary impact and potentially even a health impact. If we look, for instance, at conditions that humans have and chimpanzees don't, we could use those cell lines as a platform to study that. Lastly, it does take a village to do science. I wanted to thank this group of people that was directly involved in the work that I presented: Rusty Gage at the Salk Institute, Sarah Linker, Renata Santos, Krishna Vadodaria, Inigo, Diana, Anna Mendes, Ruth, and Jonathan. Those were the individuals that were directly involved in this work. I also would like to thank my collaborators, Caterina Semeneferi at the anthropology department at UCSD. Branka Mihiki was the graduate student that did some of the quantification of this work. Fred Gage at the Salk Institute, Bilal Kerman, Krishnan, Padma Ban, Chris Benner, Pascal Gagnon, which is also at the anthropology department at UCSD, and Ajit Farki, which was the former co-director of CARTA. And I would also like to thank this amazing group of women that worked a lot and very hard for this work to be done, and without them, there will be no work done. So Ruth, Ariana, and Anna, thank you so much, and thank you all for participating.